Shows you all the divisions of the bronchial tree, starting with division 0, the trachea, before the airway divides. You refer to the airways to their division. The first division are the primary bronchi, secondary bronchi, tertiary, all the way to the last divisions, where you have, and the numbers are pretty amazing when you think about it. You got 8 times 10 to the 7 bronchioles. It's a big number. 80 mil. 80 mil bronchioles. The airways are very small though. 0.5 millimeters in diameter. The last division is the dead end, the alveoli. Something as many as 6 times 10 to the 8. .600 mil alveoli. 0.3 millimeter. That's the dead end. And this is the, you have this many available for gas exchange with the pulmonary capillaries. Let's look at the what's called the respiratory membrane. $600 million of these, and it shows you the different cell types. The alveoli are tiny air sacs that interact with the pulmonary capillaries. And so what I want you to know is how they blow up this picture here. It shows you how the capillary is kind of stuck to the alveolus right there. So they kind of blow this up. So at the corners here are the pulmonary capillaries. And what we need to understand is how gas exchange occurs across the respiratory membrane. We got the two cells. The main cell that makes the alveolar wall is a simple squamous epithelial cell, and it's called just a type 1 cell. It's the chief cell of the alveolar wall, and it's well, I'm going to try to draw that picture there. There. Type 1 alveolar cell. Think of it as a simple squamous epithelial cell. And it's stuck to a pulmonary capillary. So the red cell is a pulmonary capillary, it's an endothelial cell. Blood vessel. They're the same kinds of cells. They're both simple squamous cells. There's two flat cells. They're stuck together with a basement membrane. It's connective tissue that kind of fuses them together. Fused basement membrane. So all three things, boom, 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 is the respiratory membrane. And this is what you diffuse gas across. You follow your diffusion gradients. So if this is the inside, this is where the oxygen leaves and is picked up by the blood. The CO2 is dumped into the alveolar space. So the CO2 gradient is the other way. And you breathe that off, CO2. So that's the gas exchange. That's the membrane. There's other cell types here besides those two. The picture shows these green cells. They're a little more round than type 1 cells. They're type 2 cells, and they're called surfactant secreting cells. Also have type 2 surfactant secreting cells. Surfactants break surface tension between water molecules that line the alveolar spaces. Surfactants break surface tension between water molecules that line alveolar spaces. By breaking the surface tension, it just makes it easier to breathe. Okay. Sometimes I illustrate the respiratory system like this, with a flask, with the narrow part of the neck of the flask being the conducting airways, but the round part being the alveolar space. And you fill these spaces with air, but the wall of the alveolar space has the type 1 and type 2 cells, but there's water that lines the inside. So imagine all these little water molecules that line the space. And they're in a circle. The thing about it is, there's cohesive forces between the water molecules, and they're kind of attracted to each other. Okay, so I'll kind of link them together. They like wanna, they're attracted to each other, but because they're arranged in a circle, it creates an inward force. So it's like, they wanna collapse the space that they're in. And you have to break that surface tension to inflate the alveolar spaces with air. So that's what surfactants do. They basically intersperse themselves in between water molecules. So surfactants, I'll use orange, maybe they just kind of shove themselves in between water molecules. And when they do that, you break the surface tension forces. It makes it easier to breathe. Okay, so that's important. I'll talk about that again. So anyways, those are the type 2 cells. Not as many of them. You mostly have the type 1 cells. The type 1 cells is the wall. The type 2 cells are there just for surfactants. There's another cell type called the alveolar macrophage. On the figure, they're colored purple. I know those as well. They're macrophages, so that's what they do. They phagocytize things, they keep things sterile in the alveolar spaces. I'm going to move on, and I'm going to turn the lights off so we can see the picture better. This is an alveolar space on the back of our model. You have some time to study the models today. And I just posted a few days ago. I'm going back to, remember I did the labels on, labels off for the first lab practical? So I'm going back to that, and I made respiratory model photos. Raise your hands if you've seen that file. Okay, so I know it's there. The rest of you, look at it, okay? Start thinking about lab practical 3. This is on it. All right, so they show all these pulmonary capillaries. They show you these star-looking cells, which is an alveolar macrophage. And look right there. They show you 2 cells. It's type 1 and type 2. How do you tell the difference? Type 2 is more round, type 1 is more flat. I will ask you that. Type 1 is more flat, type 2 is more round. That's how you tell. It's your only clue. There's no color indication or anything else. Type 1 is more flat, type 2 is more round. Just remember that. You'll be good. Here's some histology images. This is what the lung tissue looks like under a microscope. You should be able to recognize a bronchiole. Bronchiole have a very thin epithelium, say right here. Whenever you see the lining look like this, a little bit thicker, this is the pathway of a bronchiole. Okay. Now, this bronchiole divides here. Do you see how it changes from here past this point? It changes to alveolar duct. So be very careful where I point. If I point to here and I say identify, this is alveolar duct. If you go back up into here, it's bronchiole. Okay? So again, what is this hallway here? Alveolar duct. There's other things. Look at, it looks like kind of a circle. That's an alveolar sac, and the individual things here are alveoli. Okay, so I could ask you that, alveolar sac. What do you think this is? If it looks more like a hallway, it's a duct. If it looks more like circular, it's alveolar sac, and I'm going to an individual that, I'm going for alveolus. So those are the different things you have to look for. Bronchial, alveolar duct, alveolar sac, alveolus. Here's a close-up image. Identify alveolar duct, identify alveolar sac. What if I point to just that? Alveolus, very good. Let's move on. This is the lining. It's a close-up view of the respiratory bronchiole, or just bronchiole. I don't know if it's respiratory. I have to see an alveolus coming off of it. This is the smooth muscle of the bronchiole. The epithelium is it's like a cuboidal epithelium with some cilia. It's kind of sparse. So let me write that on the board. I want you to be able to identify these tissues. So we got bronchiole. be able to identify the smooth muscle. The epithelium is a ciliated cuboidal ET. Remember, the smooth muscle is important for physiology. You can dilate and constrict at the bronchial level because of it. What I did was I put the bronchial close up to the high bag of the tracheal wall, which I covered last time. You can see how thick it is in the trachea. I mean, this is showing you the mucosa and part of the submucosa. I'm not even showing you all of it. And I'm not showing the adventitia. Remember, the function of this is not for gas exchange, but just conducting airway. This was ciliated pseudostratified columnar ET with goblet cells, so it's very different. And there's no smooth muscle. There's a submucosa and adventitia with hyaline cartilage, but there's no smooth muscle. You see it in the bronchioles, though. Okay, that's one of the structural changes. So the airways are different when you get deep into the lung tissue. Let's talk about the lungs themselves. We'll take a macro view, gross anatomy, no more histology. Let's look at just the lungs. And what you see, very simply, we have left and right lungs. And just be able to identify left lung or right lung and their lobes and the fissures between them. You know, both lungs, they have an apex, okay, at the top. When lungs are inflated, the apex goes about maybe an inch above your clavicle, goes all the way up into the neck. But they both have it. I mean, that's not a way to tell. I mean, the way to tell is count the lobes. The left lung has 2 lobes, superior and inferior lobes. Be able to identify those. The right lung has superior, middle, inferior lobes. Look for those. And on the right side, those 3 lobes are separated by 2 fissures. Be able to look for the horizontal fissure and the oblique fissure. The fissure is a deep groove between the lobes. Fissure and an oblique fissure. So horizontal oblique fissures on the right. And on the left side, you have 1 fissure dividing 2 lobes. It's an oblique fissure, separating the 2 lobes on the left. They call this the costal surface because it's the side of, it's the front-facing part of the lungs that touch the inside of your chest wall. Okay. Now, if you look on the medial aspect, there's the hilum. The hilum allows things to enter and exit. This area here, it's called the hilum. There's the word there. I'll write it on the board, but you can kind of see on the left lung the cardiac impression that the heart makes. And you can even see the aorta there. Okay, you should be able to identify 3 things in the hilum on the right and the left: the artery, the vein, and the airway. The pulmonary artery, the pulmonary vein, and then the airway, which is, the main bronchus. And that's another way you can tell right from left. Because on the right side, the artery is anterior to the airway. And on the other side, the artery is above the airway, it's superior to it on the left side, okay? That's because, let's remember that how I think of it is, the artery, it goes over the airway on the left side. And to me, that's kind of why it kind of pushes the airway down, so it's underneath the artery on this side. And over here, again, the artery is anterior to the airway. All right. The pulmonary artery, that is. So on the right side, artery is anterior to airway. And on this side, artery is superior to airway. So, but on both right and left, for the hilum, be able to ID pulmonary artery, pulmonary vein, and the bronchus. Okay, so I'll just kind of put a line on it. That's for both right and left. Be able to identify hilum and stuff going in and going out. So the anatomy of lungs is pretty simple. What's more complex is their mechanics. The mechanics of breathing. So this is kind of more switching gears to physiology. And because breathing is so important, we spend quite a bit of time talking about it. You have to understand the pleural cavities to understand how we breathe. So let me kind of talk about that. I have a lot of pictures here to explain the pleural cavities. And let me read what I have here. The basic idea is to understand breathing mechanics. The most important thing to take away from lecture today is that the lung wall is stuck to the chest wall. That's really important to get. And it's not hard to understand. Two things stuck to each other, right? They're stuck together by the pleural membranes. The interactions of the lungs and the chest wall. So we just looked at the lungs. When we say chest wall, you had anatomy, right? Chest, what's your chest? Ribs, and the muscles between them, right? The intercostal muscles. So whenever I say chest wall or thoracic wall in this lecture, that's all I mean, ribs and muscle. That's the chest. So the interaction between lungs and chest wall is not by direct attachment, but through the pleural cavity, a fluid-filled space between parietal and visceral pleura. Okay, that's the first paragraph. So let's note the pleural membranes listed here. They're what's called the 3 P's of anatomy: the pleura, the pericardium, and the peritoneum. They're all serous membranes that are the linings of things. These are the linings that surround the lungs, and the pleural membranes are, there's parietal and visceral. The parietal pleura lines, as you can see from the picture, it lines the inside of the chest wall. Visceral pleura is intimate with the lung wall. It lines the surface of the lung wall. And then there's a gap in between these two membranes. It's the pleural cavity. This cavity is a very thin millimeter space between parietal and visceral pleura, and it's fluid filled. A thin, fluid filled space between parietal and visceral pleura. If you look on the picture on the middle right, it's showing you lungs, pink, and you can see the pleural membrane surrounding it. Here's lungs in the chest. The lungs are the pinkish, and you can see the blue as it extends down. So you can see that the pleural membranes occupy more space in the thoracic cavity than the lungs do. They completely envelop the lungs, and they occupy more space. The picture on the bottom is the lollipop lung analogy. If you shoved one of your Halloween suckers into a water balloon, and the water balloon completely envelops it, the lollipop or the lung, that's like what's called the pleural cavities, or what they call it here the pleural sac. Okay? So on the picture on the bottom, the part of the water balloon that's touching the lollipop lung, is that parietal or visceral pleura?
Visceral.
Visceral, excellent. Then the outer part of the balloon wall will be touching the chest wall. That's like the parietal pleura by anatomy. Let's read the second paragraph here. Because the lungs and the thoracic wall pull away from each other on opposite sides of the pleural cavity, the intrapleural pressure is less than atmospheric pressure. So let me break that down. I didn't really mention this before. The lungs and the chest wall, I said they're stuck to each other, but they're pulling in opposite directions, okay? Because the lungs are always inflated with air, and they're like a balloon. And what do balloons do once you inflate them, but you don't tie it off? If you let go of the neck of the balloon, the air wants to come out because the balloon, due to its natural elasticity, wants to collapse. And the lungs are like that. They always want to collapse, okay? So it's good that they're stuck to the chest wall. Being stuck to the chest wall holds the lung wall out. So the lungs are like a balloon. And well, think about what the chest wall is. I don't have a skeleton up here. But the rib cage is stiff and conical. Okay? It's literally a cage that naturally does not want to collapse. So it's like always pulling in the opposite direction. And so because they're pulling away from each other, it creates a negative pressure in the, we call it the intrapleural pressure. Basically the pressure in this sliver of space called the pleural cavity, if you were to measure it, it's a negative pressure with respect to the atmosphere. So this pleural cavity has an intrapleural pressure. Sometimes I abbreviate it capital P for pressure and IP subscript as an abbreviation. And it's negative with respect to atmospheric pressure. Pressure of the atmosphere. Barometric pressure at sea level. 1 atmosphere is about 760 millimeters of mercury. The pressure of the atmosphere of Earth, the air pushing down on our bodies. Sometimes it's called pressure of the body surface. We just call it atmospheric pressure here. 760. What we're saying, PIP, is negative to the pressure of the atmosphere, okay? So sometimes I call pressure of the atmosphere PATM, and it's 760. millimeters of mercury. Again, that's the pressure that can displace a column of fluid, in this case mercury. So, and typically, PIP is negative to it, which means it's a little less, so 756. It's less than atmospheric pressure. It's negative because of that. The lung wall, chest wall kind of always pulling apart from each other. Yeah, and sometimes we just kind of set the baseline 0. 0 being the pressure of the atmosphere. 0. If I'm saying pressure of the atmosphere is 0, then what's PIP? -4. It's 4 less, right? So any questions on the numbers? Okay. We'll move on. So we got the pleura, we got the pressures, and well, this kind of is a good place to talk about a collapsed lung. A collapsed lung is if you break that seal between lung and chest wall. Call it secondary atelectasis, which is the fancy way of saying collapsed lung. It's the collapse of a previous inflated lung. When did you first inflate your lungs in your life? When you're born? Took your first breath? Yeah. So they're inflated your whole life unless you sustain an injury, say like this, bullet hole in your chest, and it breaks the seal. The lung collapses. Like I said, the lung is comparable to a balloon. If the distension cannot be maintained, the lung's elastic recoil causes it to collapse just like a balloon would. If a wound opens through the thoracic cavity, air will be sucked into the pleural space because of the negative pressure, called a pneumothorax. Pneumo means air. The surface tension adhering the lung to the chest wall is broken and the lung collapses. Okay. So, basically break seal between the pleura, due to like an injury, bullet hole, knife wound. Okay, and when you break the seal, air can enter. Call that pneumothorax. If fluid enters, like say blood, when I say enters, it's entering that pleural cavity I talked about, called the hemothorax. In both cases, lung collapses because the seal is broken. What happens is air enters the pleural cavity and it equilibrates with atmospheric pressure when air enters. So if you're at PIP, -4, you're going to equilibrate with pressure of the atmosphere after air enters. Air enters, PIP is now 0. PIP is now equal to pressure of the atmosphere. It should be negative all the time, 24/7. If it equilibrates, there's nothing keeping the lung wall stuck to the chest wall anymore, and the lung completely collapses when it equilibrates. Equilibrates with PATM, which is always 0. Okay, when you get that equilibration, when the PIP equilibrates with the pressure of the atmosphere, then the lung collapses. Because the pleural cavities are separated, you have a pleural cavity, you have the pleural cavity separated from the other lung, you can still have fresh air from the other lung, but this air needs to be evacuated, and you can reinflate the lung by inserting a chest tube. Okay. You know, and I see the students, they practice. They just have a needle. They call it a decompression needle if you have to do it in the field. You just stick it in the intercostal space, and they have their spots where they're trained to stick it. And I've been told, you know you stuck it in far enough if you hear a pop. I mean, what's the pop? You're letting the air out, right? You ever like, I don't know, as your kid, like a glass Coke bottle, and you suck the air out, and then your tongue gets pulled in? You ever do that? No. Okay, well, for those of you who have, that pull your tongue in the bottle, you suck the air out, that's this. That's how it's supposed to be. That's the seal. Okay, and it's keeping it stuck. But if you kind of get your tongue out of there, break the seal, there's no force, and the lung collapses. Well, I don't know if that's going to help you understand this. This is what you got to understand right here. It equilibrates, lung collapses. You have to reinflate that. So maybe lung collapses, needle decompression. There's an emergency one where you have to do it in the field, and it's called tension pneumothorax with rightward shift. The tension that's building, tension is pressure. Say for example on this side, you have an injury to the pleura and it creates a flap that acts like a valve, a one-way flap. Because usually as you continue to breathe with this injury, the flap can open. And every time you breathe in and the flap opens, you allow air in. But then as you breathe out, the flap closes and the air cannot escape. So that's going to continually build tension over a very short period of time. I'm not sure the exact time course. But as you let air in and you do not let air out because the flap closes, that's called a tension pneumothorax. The rightward shift occurs because if it's shifting this way to the right, it's like the heart is being crushed. And you may even put a kink in the aorta. So that's one where like it's an emergency. You have to decompress as soon as possible. You can't wait till you go to the hospital to get a chest tube. Okay, that's one that I've heard about. Okay, let's move on. Here's the picture from the book about tension pneumothorax. It's a pretty good one. And what they do is they put either wound or spontaneous, but it's -4, right? It's -4. In both situations, whether it's wound or spontaneous, the side where the wound is, the lung collapses. Okay, and it's hard to inflate that. You can if you evacuate that space. I remember one semester, I had a student in 430. She had this really bad cough. It's no secret when you have a cough and you're in class, right? And this was a hacking, really. I never said anything to her, you know. But it turns out, I remember she had her from 431. I overheard her talk. He said, "Oh yeah, I had this because I was just coughing so bad. I just had a spontaneous lung collapse." So it can happen just without a serious injury like that. Collapsed lung. Think of your lungs as halfway inflated balloons. You ever like have like a really big balloon with a thick wall and it's super hard to inflate? You're like almost going to burst a blood vessel, but you finally get it inflated. Imagine that super hard, difficult to inflate balloon. Let the air halfway out, but keep it half inflated. And then take another balloon just like it. Which balloon is easier to inflate? The one that's half full or the one that's completely deflated? The half full one. It's already half full. It's easier to reinflate that. That's what your lungs are always like when they're stuck to the chest wall. If you let it completely collapse, it's like that completely deflated balloon that's more difficult to inflate because it's just completely deflated. So the idea that your lung is stuck to your chest wall is a very important idea. Keeps your lungs easier to inflate. So let's learn about the muscles of breathing that help us inflate the lungs. The main one is the diaphragm. Diaphragm. That's your main muscle of inspiration. And you look on the picture on top right, it's a big dome-shaped, thin, membrane-like muscle. Remember, diaphragm means membrane. See how like far up into the thoracic cavity it goes into? I've seen this dissection. Like you can stick your head up there, no problem. It's a big space. Okay. The picture on here shows you contracted, relaxed. When the muscle is relaxed, that's the blue. It's more domey. The muscle flattens as it contracts. Flattens during contraction, while on the top left, the red. And what you're doing is, if you look on the inferior view, looking up into it, right here, you can see that all the muscle fibers are arranged circularly around this white central tendon. So they're pulling in a circle, okay? So when each muscle fiber pulls, the thing is pulled down, okay? Fibers pull down on central tendon. Because it flattens, if you look at the top left, you're actually creating more thoracic volume when it contracts. That's the main idea here. When you contract, you increase thoracic volume. That's important, because if you increase the volume, you decrease the pressure. Increase volume, decrease intrapulmonary pressure. All right, so I'll get into more of that later. That's the main thing of how you breathe. You have to change the pressure inside the pulmonary spaces to draw air into it. But more anatomy here. There are 3 orifices in the diaphragm you're supposed to know. There's a hole here, here, and here. 3 holes, each for inferior vena cava, esophagus, aorta. Okay, you need to know those. Let me write them here. So you got the orifice for the inferior vena cava is called the caval opening. It's the highest one up. It's for inferior vena cava. And if you look at the picture, if you count the vertebra, it's at around the level of the T8 vertebra. The next one is the opening for the esophagus. So it's called esophageal opening, and it's about the level of T10. The opening for the abdominal aorta is at about the level of T12. So that's where the name of the aorta changes from thoracic to abdominal aorta. I've already talked about aorta. Well, anyways, that's the diaphragm. Helps you breathe mostly, that's its main function. There are other muscles that help you breathe, and I put this picture there. The muscles between the ribs, between each intercostal space, there's the external and there's the internal intercostals. Along with the diaphragm, the external intercostals and the diaphragm are considered your muscles of inspiration. They help you breathe in because the external intercostals, they basically elevate ribs. The rib is a curved structure, like a bucket handle. And when you elevate them, it's like lifting the handle of a bucket handle. Okay. Called bucket handle movement, and I'll show you a picture of that later. But basically, elevate ribs, along with diaphragm, you're expanding your chest wall, okay? Again, the idea is increase thoracic volume, decrease intrapulmonary pressure, I already wrote it. But there's other muscles. There's the internal intercostals, they depress ribs, so that helps collapse your rib cage. So, if diaphragm and external intercostals are for inspiration, depressed ribs would be for forceful expiration, huffing and puffing. Because normally, during normal quiet breathing, a breath out is a passive event. You don't need to recruit muscles for that. Just let the elastic recoil of the lungs take over and let the air out with no muscle recruitment. But if you're really huffing and puffing or you have labored breathing, you do recruit muscles like these ones. And actually, any muscle that has an attachment to ribs can be considered an accessory muscle of breathing. Any muscle with attachment to ribs can be considered an accessory muscle to breathing. You can almost include clavicles too. Well, anyways, it's not shown here, but the sternocleidomastoid can be considered one. Pecs, pec major, pec minor, serratus anterior. I'm just blurting out the ones off the top of my head. Scalenes, anterior, middle, posterior. Okay, just to name some. There's others, but that's the concept. Attach the rib, you can move rib cage, you can help breathe. So I want you to know how inspiratory muscles affect the rib cage. When the chest expands, how does it expand? Think about your sternum. The joint between the manubrium and the body of the sternum is called the sternal angle. That can pivot just a little bit like a pump handle. So they call this the pump handle movement of the sternum. Expands the chest wall front to back just a little bit. So that's what this figure shows you there. I'll call it movement of sternum. At the sternal angle is like a pump handle movement. This is how you expand the chest during your breath in from front to back, from anterior to posterior. Expand chest, anterior to posterior. From front to back. I left it up here. I said before, when you elevate the ribs, it's like a bucket handle movement. Okay, when you lift the bucket handle, they even show you a little bucket handle there to show what's meant by that. The ribs are lifted up just a little bit. That's just enough to expand the chest laterally from side to side. So this movement, this bucket handle movement expands, expand chest laterally. So all that to talk about more chemistry, because you can change the volume, you change the pressure of the gas, because we're talking about air. Gas, air is a gas. And most chemistry students, you remember studying this equation, right? P1V1 equals P2V2. If you've forgotten, let me remind you, it's really simple. One thing to understand when you talk about gases, not liquids, gases are different from liquids. If you have a cup and you fill it halfway with a liquid, it stays half full or half empty. But the half full liquid doesn't expand and fill the glass that it's in. Gases, it does do that. If you have a closed canister and you insert your gas molecules, they fill the space that they're contained in the gaseous state. And those bouncing around gas molecules exert a pressure on their container that you can measure. So if volume is 1 and pressure is 1, great. If you change the volume, you double the pressure. If you change the volume to half, you increase the pressure by 2. That's the equation. That's the relationship. Okay? So with breathing, the physics of the lungs, airways, chest wall, and how they change in lung volume, and how they cause changes in lung volume, which creates pressure gradients to move air in and out of the lungs. Okay? So that's what we're talking about now. And to understand that, let's review our pressures. When you study this, keep track of 3 pressures. Pressure of the atmosphere is 760 or 0. Let's remember that intrapleural pressure, the PIP, is always negative. And let's remember that the intrapulmonary pressure, I'm going to abbreviate it P-pole. Pressure inside the lung. Okay, now the P-pole, sometimes called, the intraalveolar pressure. It's the pressure literally inside the alveolar space. Now, I can't put a single number to it. I mean, it changes with breathing. It changes between -1, 0, +1. Millimeters of mercury. It changes with breathing. Okay. So what we have to do is, if you're gonna like move air in and out of your lungs, you have to create a difference between the pressure of the air you're breathing, ATM, and pressure inside your lungs. They have to be different for air to flow, okay? And so, that's why we have this down here, the transmural pressure. We talked about that before in blood pressure. Now we're talking about it with movement of air. This time, transmural pressure is the pressure across the whole system between your lung wall and your chest wall, okay? I think I have a picture of that there. So let's call this P-trans just like we did before. Transmural pressure. Pressure inside the lung, P-pole, versus pressure of the atmosphere, which is always zero, barometric pressure. And it's always pressure in minus pressure out. So P-trans equals, like I say on the top there, P-pole minus pressure of the atmosphere, which is always going to be zero. So you just have to change the pressure inside your lungs so air will flow. And so now we talk about the forces that make the chest wall expand and collapse. We have to talk about both the lung and the chest. And what I've been saying all lecture is the lung is like a balloon. That force, we call it in. Collapse is like an inward force. So lung forces versus chest wall forces. Lung forces are in. I said the lung is stuck to the chest, but that they pull in opposite directions. If I'm calling lung forces in, what do you think I'm going to call chest wall forces? Out? Yeah. They're pulling against each other. Forces acting on the chest wall, they're out. They make the chest wall is like out because it's a stiff cage. So think of it this way. The lung holds the chest wall in, or different way of saying the same thing. The chest wall holds the lung out. They're stuck to each other. And so the forces that make the lung collapse in, you have to think of the anatomy that I've been talking about. There's a lot of elastic tissue surrounding these round spaces. So again, remember I drew the alveolar space like that. Remember how I talked about how you have the water molecules and there's surface tension between them, but you can have surfactants that help reduce that surface tension. That's great. But there's still surface tension. So one of the things we have to consider are the surface tension forces that make the lung collapse. ST. Surface tension. But if you look at the picture, surrounding it are elastic fibers. So it's like if you take, rubber bands and wrap them around. That it like wants to collapse it. So they call that EL. The elastic recoil. So on the bottom, let's talk about the forces of the chest wall. I mean, it's a stiff cage. So the elastic recoil forces they're just out, okay? It's just elastic recoil, but of the stiff cage, it's a force out. We just say the elastic recoil due to the elastic fibers here is a force in, makes it collapse. So you got rubber band fibers collapsing in lung, but you got the stiff cage is an elastic recoil in the opposite direction. So it's out versus in, and the question is, which force is greater, the force out or the force in? So even if you haven't taken physics, that's not hard to understand. Out, in. Well, they're pulling on each other. The analogy has been given, like if you put two glass plates and they're separated, you put a small drop of water on one, then put the two glass plates on top of each other, you can slide them back and forth, but when you try to separate the glass plates, that little bit of liquid keeps them stuck, just like this. And imagine a spring on either side, one's pulling in and the other's pulling out. I remember this analogy, my physiology professor gave it to me, and he was telling us like, well, if the chest wall forces are greater, you're gonna pull the chest out, like in a breath in. And if the lung wall forces are greater, you're gonna take a breath out, your lungs collapse and everything collapses. So breathing in and breathing out is simply whether the chest wall or the lung wall forces are greater. That's all it is, like springs pulling on each other. Yeah. And so when we consider breathing, there's two parts. Well, actually there's three. When you take a breath in, it's followed by a breath out. Then there's a momentary pause before you take your next breath, and you breathe at a certain rate. The average person will breathe like 12 to 15 breaths a minute. Okay, and, I mean, as a clinician, you'll be asked to record patients' respiratory rates, RR. Okay, well anyways, let's consider one breath and consider the pressures and forces acting on the lung chest wall system during breathing. There's something I haven't defined yet. It's FRC. Let me define what that is. Hold on. Number one, FRC. That stands for Functional Residual Capacity. You don't even have to write it out now because I'm going to give a whole lecture on it next week. But just for now, note that this is before you take your next breath. It's that momentary pause between breaths. Think of it that way. Okay. This is before you take the next breath. So what I say on the slide is nothing moves. You haven't done anything yet. Breathe in, breathe out, pause. Before you breathe, nothing happens. That's why I say nothing moves. Okay? So basically what that means is the force of the lung and the force of the chest wall pulling in and out is equal, so nothing moves. When forces are equal, nothing moves. Okay? A good balloon analogy is when you blow up a balloon, and say you tie off the balloon, and this balloon just stays there. Okay, it's like the balloon wall, it has been inflated, but it doesn't move because you've tied it off. Look, it's kind of like that. That's why I put balloon analogy there. Nothing moves. But of course, when you breathe, you want things to move so you can ventilate air in and out. So, at FRC, think of it this way. You need to consider the transmural pressure P pull minus pressure of the atmosphere, and you have to consider forces. Lung, chest wall. Forces are equal at FRC. Okay. In terms of P-trans, pressure of the atmosphere is always going to be 0. And I'm going to drop the units, the millimeters of mercury. But I'll just tell you that before you take the next breath, the pressure of the atmosphere is equal to P-pole. They're both 0. So the pressure across the whole system, P trans, is 0. 0 minus, 0 minus 0 is 0, right? So what that 0 means to you, what 0 means is there's no gradient, so air does not flow. That's what 0 means. There's no gradient. There's no difference between inside, outside. No gradient, no airflow. But of course, in breathing, we want air to flow, so that's inspiration. So let's kind of talk about the real part of breathing. So to expand the lungs, we need the expansive forces of the chest wall to be greater than the recoil forces of the lung. You have to expand the chest wall. So you got to use your breathing muscles, and I list a few there. So I'll do inspiration. Take a breath in. You have to recruit breathing muscles, muscle recruitment. Recruit muscles of inspiration. And I list them there. Diaphragm, external intercostals are the main ones. Okay, scalenes, yeah, not so much. Really, the first two. Diaphragm, external intercostals are your main muscles of breathing. And what you're going to do is, those muscles will expand the chest wall. You're going to increase thoracic volume. And as a result, you're going to expand the lung, and you're going to decrease P-pole. So in our forces, the lung wall forces are greater, okay, greater than the chest wall. So things expand, and the pressure inside the alveolar spaces will decrease from 0 to -1. And the pressure of the atmosphere is still 0. So when calculating the transmural pressure, and you don't need a calculator for this one, right? The math is simple. -1 minus 0 is -1 millimeters of mercury. That means the space inside the lung is negative with respect to the outside. So air flows in. That's what -1 means. As a result of this -1, air flows in until you equilibrate to 0. So it goes, breathe in, expand the lung chest wall, you create a negative pressure, air flows in until you equilibrate to 0. So at the end of a breath in, it's equilibrated. At the end of a breath in, okay, and that is to draw air into the lungs. So there's an old saying, we breathe by negative pressure. Okay, that's how we breathe. We create a negative pressure and air flows in. Oh, basically before I show you that, some anatomy here. Side note. You see how you have rib and then right under it, intercostal nerve, artery, and vein? If you ever insert a chest tube, and I'm sure they'll train you in this, it's better to insert the needle at the superior border of a rib, not the inferior border, because you'll damage that. Okay, since I got that close up there. Just mental note. All right, well anyways, okay, this thing here. All right, so imagine you're going to take a breath in, you're at FRC, P-pole is 0, then you expand the lung wall because the chest wall has expanded, so you make the space inside greater so that the air molecules, gas molecules, they fill their space. But because you've increased the volume, you decrease the pressure, just a little bit, right? That's enough to create a gradient for air to flow in until what? You equilibrate to 0. Okay. So once again, before you take a breath in, you're at 0. First, you increase the volume, decrease the pressure, air flows in until you're at 0. All right, so let's take a breath out. Normal quiet breathing is what you're doing now. You're not exercising. We've exercised in this class. I want to mention why I got exercise on my mind. It's not on the schedule, as a little side note. For Monday, because I'm a little ahead of schedule, I want to put the gas exchange lecture for Monday. Because right now it's on the schedule for Friday, but because I'm ahead, I'm going to switch that to Monday, and we'll keep Wednesday the same. So this lecture involves a brief lab activity, and it's a new one, it's not on the schedule, and it requires someone to exercise. So a little bit of lab activity on Monday. On Wednesday is your mandatory BioPath lab. So I just wanted to mention that so someone comes dressed to exercise. All right, back to this. Inspiration was active, expiration is passive. You don't recruit muscles. You don't need to in normal quiet breathing. Basically, muscles relax and the lungs recoil. So there, oh wait, oh shoot. I think I messed up. Dang it. I'm gonna go back. Hold on. Okay, look at your notes. Which forces did I say were greater? I messed up. Reverse that. For inspiration, chest wall forces are greater. So just reverse that arrow, just cross out the greater and put it under here. That's why the chest wall expands. Okay, now, expiration. Muscles relax, lungs recoil. So, okay, that's the mistake. Sorry about that. So inspiration, chest wall forces are greater. Now for expiration, no recruitment of muscles, system collapses, lungs are greater. So this is correct. So, because the thoracic volume decreases when your chest wall collapses, you increase the intrapulmonary pressure. Okay? So the P-pole changes to +1. And the pressure of the atmosphere is still 0. So all you do is, that simple addition is, the P-trans is +1. And the +1 means that the inside of the lung is positive with respect to the outside. So air flows out until you equilibrate. It flows out until you equilibrate back to 0. So you're just changing it from +1 to -1. So if you, at the end of the breath in, you equilibrate. In the breath out, the lungs collapse, and then the alveolar spaces shrink, increasing the pressure from 0 to +1. That's a gradient to push air out for expiration, and you do it until it equilibrates to 0. So that equilibration at the end of a breath in and at the end of the breath out is shown in the data on the top slide. Okay, so this right here, it's like a sine wave. Basically, when we talk about the mechanics of breathing, you should correlate your notes with this figure here. And so this is where I was talking about breath in, breath out. You see the green dot over there? Right here, following. You're at FRC. Everything is not moving and you're at 0. So when you take a breath in, you expand the chest wall and the lungs expand with it, and you create a negative pressure. -1. Air flows in until you're at 0. Then when you take a breath out, you're at 0. The system collapses and you create a positive pressure. Okay? And so you go to +1, follow the green dot. The air flows out until you equilibrate again. So you're always equilibrating. You equilibrate at the end of a breath in and at the end of the breath out. That's the main idea there. Okay, so lung compliance. With surfactants, it's a lot easier to breathe. You only have to generate 1 or 2 millimeters of mercury difference to get air to flow in and out of your lungs. Without surfactants, you would have very labored breathing. You'd have to generate a much larger gradient. Okay. All right, so let's review this since I kind of messed it up. Let's see if you can answer this. Turn the lights on. I talked about lung and chest wall. Which forces dominate at FRC? Or are they equal? They're equal. Which forces dominate during inspiration? It's the chest wall. And what about expiration? The lungs. Okay, so that part's straight. And I'll show you this after, but let's take a break and we'll finish these slides when we come back.